Hello, this is Priyadharshana. I am working as an assistant professor of Botany, Standard Firework, Rajaratnam College for Women. Today we are going to discuss about the mechanisms of crossing over. So coming to the definition, so crossing over is the process of exchanging of genetic material or segments between the non-sister chromatids of two homologous chromosomes. So non-sister chromatids means the chromosomes having the same length are termed as non-sister chromatids. Crossing over occurs due to the interchange of sections of homologous chromosomes in the packaging stage of prophase 1. The crossing over takes place at the 4-chromatid or tetrad stage of meiosis. This crossing over theory was first described by Thomas Hunt Morgan. So, there are 4 stages of 4 mechanisms in the crossing over. The mechanism or process of crossing over includes the following stages: synapsis, duplication of chromosomes, crossing over and terminalization. First one is synapsis. The paternal and maternal chromosome of a homologous pair come close together during the prophase 1 of meiosis cell division. They pair at the zygote substage, which is called as synapsis. During synapsis, the homologous chromosomes lie side by side and coil around each other to form an bivalent. So, here the two chromosomes comes too close together, so this is called as synapsis. So, duplication of chromosomes. So, this is the second mechanism. Synapsis is followed by the duplication of chromosomes. Here, each of the homologous chromosomes in a bivalent splits longitudinally into 2 sister chromatids during diploid stage. Thus, the bivalent now consists of 4 chromatids, which is called as tetrad. So, here we can see this diagram. So, in each chromosome we are having the 2 chromatids, it duplicates and it forms 4 tetrad stage. So, third mechanism is the crossing over. During packaging stage, when the paired chromosomes start separating, the chromatids remain attached at one or more points and thus establish one or more exchange per bivalent. This point of attachment is known as chiasma. At each chiasma, 2 non-sister chromatids of the bivalent break at the corresponding points and then rejoin with the exchange of segments, forming a new chromatids. So, likewise crossing over takes place. The 2 chromosomes comes closer and they start to pair. So, last mechanism is terminalization. Here the non-sister chromatids start repelling each other during diakinesis substage after the completion of crossing over. The chromatid separates from the centromere. The chiasma shift towards the terminal ends. The movement of chiasma towards the end is known as terminalization. Gradually the chromatids get condensed and thus shortened and the homologous chromosomes become separated. So, this is the final stage. So, the 2 chromosomes pair and it becomes a recombinant chromosomes. So, these are the mechanisms of crossing over. So, first they comes closer and they start pairing and finally we get the recombinant chromosomes. So, when does the crossing over usually takes and what is the frequency? So, the frequency of the crossing over depends upon the distance of genes between which crossing over occurs. The frequency of crossing over between any 2 linked genes increases with the distance between them. Similarly, the closer the 2 genes are linked, the lesser the chance for chiasma occurring between them. So, these are the frequencies of crossing overs. Thank you for visiting this video. Thank you all.